So we know that allogeneic transplantation is the only curative treatment option for older patients with high-risk MDS and AML, but unfortunately, relapse is the main cause of transplant failure and death for these patients. So with my mentor, Dr. Garcia, at Dana-Farber, we aimed to augment or improve conditioning chemotherapy by adding Venetoclax to that pre-donor stem cell infusion chemotherapy, and then, assuming patients did well after the transplant, to give them further relapse mitigating treatment in the form of Azacitidine and Venetoclax as maintenance for approximately 1 year after transplantation. So in this very high-risk cohort, 50% of whom had biallelic TP53 mutations, which we know confer an exceedingly high risk of relapse, the 1-year survival was approximately 60%. Relapse remained the biggest factor that led to death, unfortunately still, in about 50% of patients. But overall, we found that it was feasible and safe to give this augmented RIC conditioning and post-transplant maintenance. We did note infections, which have been noted with post-transplant cyclophosphamide-based GVHD prophylaxis. And we also found that compared to our TAC-methotrexate cohort, there was a trend to taking a little bit longer to get to maintenance, and that a slightly fewer percentage of patients were able to get to maintenance. Clearly, these results need to be validated in randomized trials to see if this is something that is feasible, safe, and effective in the real world. There are ongoing efforts through cooperative groups, MyeloMatch, to test these hypotheses and hopefully bring this forward into standard of care if it proves to be efficacious.